Okay, so welcome to segment 2 of the protists. We're going to start moving into the major supergroups of protists, starting with the excavates. We look at this phylogeny of the protist once again, and then these are some images of some examples of protists showing how diverse they actually are. You can see that, of course, all protists have some ancestor, right? So there's a common ancestor of all protists, and also it's the same common ancestor between protist and plants, protist and fungi, and protist and animals, and then each one of those three along with each other. So here we're starting with the excavates. So we're going to explore, this is a very short segment, we're going to explore the excavates, which divide into three categories: the Diplomonads, the parabasalids, and the Euglenozoan. Now, if we look at the four supergroups, I kind of explained that when we discussed the phylogeny in the first segment, but there are four major supergroups, which then, of course, further divide into subgroups and sub-subgroups. We're going to be dealing with the excavates, but the next three segments will include the SAR group, the Archaeplastida, and the Uniconta. Now, I'm going to show you just a little brief thing to get a concept map started, and then we'll discuss all of the characteristics of it. So I'm going to go ahead and get a concept map started. However, I'm only going to do the concept map in segments. So, of course, we're starting with the excavates. Now, I want to kind of put into perspective, remember, we have a common ancestor of all eukaryotes, really. And then we have the four major supergroups. I'm going to kind of do this. I'll get this started for each one, but I'm only going to basically expand out for each of the segments the segment that we're dealing with, or the supergroup that we're dealing with. So here we have the excavates. Okay, and then the SAR group, which will be the next segment, the Archaeplastida, and the Uniconta. Okay, again, these are the subjects of the other three segments right here. Okay, we're only dealing with the excavates. So if you're going to make a concept map, you can kind of design it like a tree, and the excavates further divide into three categories, right? So that includes the Diplomonads, the parabasalids, and the Euglenozoans. Now, these are the three that are shown in the tree, right? So why replicate the tree? Will some of them further divide even more? So the Euglenozoans actually divide into two additional categories, which you would want to connect to the Euglenozoans. So remember, these are the three subgroups of the excavates. Okay, so then we have the Kinetoplastids, and we have the Euglenids. E-N-I-D-S. Okay. So that kind of gets it started. Now you want to kind of expand upon this, and I'll do each of the segment as with the discussion of the corresponding segment. But here I got this started, but what you want to do is maybe put the characteristics of each one, and maybe connect the characteristics, or maybe add the characteristics to the line that leads to that particular organism. It depends on how you prefer to make your concept map. Now, this is really one of the best ways to learn the major groups and their specific characteristics, and know which ones are related. So the excavates are quite interesting. However, all protists are quite interesting, but the excavates actually have modified mitochondrion, and they also have a unique flagella. Excavates are generally characterized by their cytoskeleton. Some members have an excavated feeding groove on one side of the body. So it's kind of like an indention on one side of the body. They generally have two flagellas, one short one and one long hairy flagella. The excavates include these three groups, and they're all monophyletic at this point. So we have the Diplomonads, the parabasalids, and the Euglenids. Now, the Diplomonads and the parabasalids are the ones that actually have the modified mitochondria, and they're a different name for each one. They do not have a plastid, which is quite interesting, and most of them live in anaerobic environments. So these are, of course, environments that lack oxygen. The Diplomonads have a reduced mitochondrion called the mitosome. They get their energy from anaerobic biochemical pathways, so they're anaerobic organisms. They have two equal-sized nuclei and multiple flagella, so you can see that in this example here. Often they're parasitic, for instance, the Giardia intestinalis, which infects humans. Okay, so back to the Diplomonads and the parabasalids. Again, we already talked about the Diplomonads. Now we're going to move into the parabasalids. Again, they still lack plastids, and they have a modified mitochondrion. However, in parabasalids, the modified mitochondrion is called a Euglenozoan. And this generates energy. Some of the energy is anaerobic. And then, of course, so they're not purely anaerobic organisms. One example is the following. So you can see the multiple flagella, the Trichomonas vaginalis. So this is actually a protozoan that is sexually transmitted. It's a parasite that's sexually transmitted. It does have a unique characteristic in this right here. Now, the Euglenozoans is the next category. This is quite a diverse clade, which includes those protozoa that can be predatory and heterotrophic, those that can be photosynthetic and autotrophic, those that are mixotrophs, and those that can be parasitic. So there's really nothing unique as far as their nutritional categories and or lifestyles. So they are quite diverse in that particular category. Now, the main feature that distinguishes the Euglenozoans from the other clades is that they have a spiral or crystalline rod inside their flagella. The clade includes the Kinetoplastids and the Euglenids. Okay, so this is the one that actually divides into two major categories. And again, the Kinetoplastids and the Euglenids are expected to also have the spiral or crystalline rod inside their flagella. So that's a key characteristic of Euglenozoans. Okay, so let's talk about the Kinetoplastids. The Kinetoplastids are quite interesting. They have one single mitochondrion. Now, you might think that all cells have one mitochondrion. No, that's not actually true. They have hundreds sometimes of mitochondrion. But this particular organism has only one mitochondrion with a mass of DNA that's inside it called the kinetoplast. Now, kinetoplasts can be free-living, and they're often consumers of prokaryotes, and they live in environments like freshwater and marine, so aquatic environments, and moist terrestrial environments. Now, some of them can be parasitic, and they can parasitize animals, plants, and other protists. So one really cool key example is the trypanosoma. Now, there's two trypanosoma species. One of them causes African sleeping sickness, and the other one causes Chagas disease in humans. So what they look like, you can see that they have this undulating membrane. They have a flagellum. And you can see that kinetoplast at the very tip of their cells. And then this is the nucleus right here. They infect blood. So they do not get inside the blood cells. They infect the blood as a whole, the whole solution, the blood system. So you can see them around the blood. Now, a little bit more about kinetoplastids. They actually can evade the host immune system. And they do this by switching their proteins on the surface of their cell in which the immune system would normally recognize. So if they switch it, then the immune system cannot recognize it. And this is done by chromosomal rearrangement. And we'll kind of talk about this later on at the end of the semester in chapter 21 when we talk about changes in genomes. Now, again, this actually allows the organism to prevent the host from developing an immunity from diseases in which it causes, like Chagas disease and African sleeping sickness. Euglenids, they have one or two flagella that emerge from a pocket just at one side of the cell. So here we can see one flagellum. Now, often Euglena have, or often they have two flagella, but one of them's not really obviously visible. Here we have a short flagellum and a long, hairy flagellum in the Euglena. Now, some of these are actually mixotrophs, so it's key to know that Euglena specifically, which is a euglenid, is a mixotroph. So they can switch between autotrophic and heterotrophic, and of course utilize light for energy and some when they're autotrophic. And it depends on their conditions in which they're in, so they can kind of adapt to their environment a little bit better than most other protists. Now, this is a key summary of the excavates. So remember, the excavates include the Diplomonads, the parabasalids, and the Euglenozoans. And the Euglenozoans further divide into the Kinetoplastids and the Euglenids. And these are some key characteristics and some examples, which may be useful to fill out your table. All right, on to the next segment.